Good morning, students. Today we can start the heredity and evolution. At the last period, we can see what is meant by protein synthesis, what is meant by central dogma, what is meant by translation. You know very well the protein synthesis means the process of formation of proteins. The process of formation of proteins from DNA with the help of RNA. The process of formation of proteins from DNA with the help of RNA, that is known as protein synthesis. The process of formation of proteins from DNA with the help of RNA or through RNA, that is called as protein synthesis. Now we can already see what is meant by transcription. The formation of RNA from DNA, that is known as transcription. That is known as transcription. Now the type of RNA also known, that also we can see the mRNA, rRNA and tRNA. With the help of that all RNA, we can form the proteins molecules. Now, how the mRNA is formed and how the proteins molecules are formed, today we can see. The mRNA formed in the nucleus comes in cytoplasm. Firstly, you know very well the DNA is present in the nucleus and mRNA takes the information from DNA. mRNA takes the information from DNA. That's why it is mRNA also present in the nucleus that takes the information from DNA, that present on DNA. mRNA takes the information that present on DNA. That means mRNA formed in nucleus and comes in cytoplasm. Nucleus मध्ये form होता आणि नंतर तो कुठे येतो cytoplasm मध्ये, just like that. Here DNA is present, the mRNA is formed in nucleus and comes in cytoplasm. Here he comes in cytoplasm. It brings the coded message from DNA. It takes the coded message from DNA. It takes the all coded message present on DNA that is takes by mRNA. Okay, just like that. You can see already that. In the form of codons. In the form of codons. A, U, G, like that. In the form of codons. So, it brings the coded message from DNA. The message contains the code for amino acids. That message contains the code for amino acids. That message contains the codes for amino acid. And that is called as triplet codons also. That is called as also triplet codons. The code for each amino acid consists of 3 nucleotides. The codes of each amino acid consist of 3 nucleotides. For example, A, U, G, adenine, uracil, guanine. The codes of each amino acid consist of 3 nucleotides. That is known as triplet codon. That is known as triplet codons. Okay. The codes for each amino acid consist of 3 nucleotides. It's called as triplet codon. It's called as triplet codons. Now, Dr. Hargovind Khurana, a scientist of Indian origin, has made an important contribution in discovery of triplet codons. Dr. Hargovind Khurana. He is the Indian scientist. He is the Indian scientist. He was an Indian scientist, and he discovered the triplet codon. He discovered the triplet codons for 20 amino acids. How many? The 20 amino acids. Different types of 20 amino acids. Each amino acid contains 3 nucleotides. Each amino acid consists of 3 nucleotides. Okay. So For his work, he has been awarded with the Nobel Prize in 1968 along with the two other scientists. He was awarded for the Nobel Prize in 1968. So, each mRNA is made up of thousands of triplet codons. See again, that is the most important. Each mRNA is made up of thousands of triplet codons. And each triplet codon made up of 3 nucleotides. Each mRNA is made up of thousands of triplet codon. And each triplet codon is made up of 3 nucleotides. And as per the message of mRNA, amino acids are supplied by the tRNA. The message contains mRNA that amino acids supplied to the tRNA. The shape of tRNA you can see like that. Okay. Like that. That is the shape of tRNA. mRNA have codons and tRNA consists of anticodons. That is the complementary sequence of codons, anticodons that compactly arranged in between them. Codons, mRNA consists of codons and tRNA consists of anticodons. Okay. So, as per the message of mRNA, amino acids are supplied by the tRNA. For this purpose, tRNA has anticodons. tRNA has anticodons having the complementary sequence to the codons of mRNA. This is called as translation. That is called as translation. You know very well, that is known as translation. See again, what is the complementary sequence of mRNA? That is like that. Compactly arranged, and between them A, U, G. A is always bonding with thymine, uracil, cytosine, guanine, uracil with cytosine. And like that. That is the complementary sequence of anticodon consists of tRNA. Okay? That is known as translation. That means only the information contained by mRNA, the information consists of mRNA that only with the help of tRNA that translate, identify that information. That's why that process is known as translation, and the definition is, as per the message of mRNA, amino acid is supplied by the tRNA. For this purpose, tRNA has an anticodons. tRNA has the anticodons complementary sequence that codons of mRNA, that is called as translation. That is called as translation. The amino acid blocks by tRNA are bonded together by the peptide bond. That bonding takes place with the help of peptide bond in between amino acids and tRNA. mRNA and tRNA, the bonding takes place, and that through the peptide bond with the help of RNA. That peptide bond formed with the help of RNA. RNA is the ribosomal RNA. mRNA messenger RNA, tRNA transfer of RNA, and rRNA ribosomal RNA. You can see that all processes. See, during this process, the ribosome keeps on moving from one end of the mRNA to another end of the mRNA. Like one end of the mRNA to another end of the mRNA. That is moving takes place of tRNA. One end of the mRNA to another end of the mRNA. Codons, triplet codons, that is known as translocation. Just one place to another place, moving of tRNA, that is known as translocation. That is known as translocation. Okay. So end of the mRNA to the another end of the triplet codons. Another end of the triplet codons, that is called as translocation. Okay. See again, transcription, formation of RNA, form DNA, that is known as transcription. Translation, the information, the code present on mRNA that compactly that read by the tRNA with the anticodons. The codons present on mRNA that read with the help of tRNA with the help of anticodons. Okay, complementary sequence of anticodons, that is known as translation, and with the help of RNA, that transformation or movement of tRNA from one triplet codon to another to the next. That movements of tRNA from one triplet codon to other end, that is known as translocation. That is known as translocation. Okay. So such many chains comes together to form a complex proteins. Such many chains comes together to form a complex proteins, and these proteins control the various functions of the body in living organism and their appearance too. You know very well, proteins plays an important role in the living organism, that's for growth, for development, and all life processes also controlled with the help of proteins. That means all life processes like energy formation, digestion, respiration, then again reproduction, that all process having a necessity of the protein synthesis. That means that proteins control the all functions of the body and appearance too. On the basis of that, DNA, recombinant DNA technology or protein synthesis, appearance, how the animals show that transfer of heredity from one generation to another generation, that appearance also controlled with the help of that protein synthesis. Okay, so today we can stop here, and again if you have any questions, so ask me as per your